How y'all doing? Yeah, it is time. It is time. It is time, in fact. Do I need to care? Okay, let me just let them know. Alright, time is like such a bad idea. Alright, let's do this. Wait, wait, wait. Just hear me out for 1 second. Oh sh. No, this is too much power. I need to. God, my mic is off. It's such a awful idea. Alright. I have to like. Oops. I have to like complete a lot today. Um, I don't know to what extent they want me to know this, right? Because like our oxidative phosphorylation was frankly pretty lackluster. It wasn't much, but they might be asking for something a bit more on the nose. So obviously I'm gonna have to lock the freaking. It's okay, man. Even if I don't get on, I don't care. Shut up. Is everything okay? Honestly, I would love to be a bit more stretched. Just a bit more stretched, but okay, this is annoying. This is really annoying. That's fine. Wait. Why? Why are you doing that? That's not what I want to do. I want to do this, but it won't allow me to. Okay, no problem. Transition. No, not transition. Transform. Oi, not like that. Uh, okay, that's better. Better. Okay. This is so dumb. This is so dumb. This is so dumb. Oh, this is so stupid. Okay, shut up. Okay, let's turn on the timer. Okay, so right now we're just doing outer structure. I already went through like prokaryote and eukaryote, like animal cell eukaryote cell structure. Not too bad. Mainly just had to remember the function of like Golgi apparatus and like endoplasmic reticulum, the smooth and the rough. Like that whole like flow chart of like what happens when the moment mRNA is sent out, as well as kind of getting a feel for what ribosomes are. That actually I'm not too sure. I kind of forgot. Minute structures. two subunits. Some are attached to the surface of the rough endoplasmic reticulum. Decompose the protein and RNA. They have two subunits. Function is they're the cytoprotein synthesis. Okay, so get the idea. This is very small. That's the only problem. Centriole as well. Lysosome as well. But lysosome is mainly used for apoptosis or autolysis. Digest the materials of the content. Digest worn out parts of the cell. Releases the enzymes outside of the cell by exocytosis. So let's see. Do I look? Okay. Nice. Okay. So just a rough idea. We start from the membrane, which is going to have a membrane and a cell wall on the outside. You're going to be having pores in the cell wall meant for plasmodesmata. That's going to be communication between the plant cells, right? Opening the cell that functions cell-to-cell communication. Then we have a large vacuole. This is the special bit. You have chloroplast. That's the special bit. Mitochondria stays because we have membrane-bound organelles, right? So you have mitochondria. You have ribosomes, of course. You have your rough endoplasmic reticulum. You have your smooth endoplasmic reticulum. You have your Golgi apparatus. You have your okay, we have a cytoskeleton. This is used for I'm pretty sure it's meant in the cell like dividing, because I know there's like a cell plate and stuff like that. I'm not really sure what that is. Peroxisome, don't know what that is. Chloroplast containing thylakoids. Organelle containing thylakoids, the site of photosynthesis. I've never heard them say thylakoids. I've heard them say chlorophyll, but I've never meant. Okay, yeah, sure. Anyways, I mean the thylakoid membrane contain, or no, what is it? I mean it is right. The thylakoid membrane contains the photosystems, and then the photosystems contain various like various amounts of different. It contains various chlorophyll pigments. Why does this not have a number? Plasma membrane, cell wall. There's also the, what do you call it? These things, which you're like guard cells and stuff like that. But I guess that's not really that's not too important. Central vacuole, storage compartment for water, sugar, ions, and pigments. And tonoplast is the membrane surrounding the central vacuole. Nucleus is the same. Okay, I'm really excited to like learn about like the xylem phloem and like the root of the cell, and learning about like the thingy, like that thing where you have like your xylem, you have your no, not the xylem. I forgot what the thing what was, but it's like the protective cells, and then you have like the phloem, and then you have the xylem, something like that, and that's like surrounding the matrix or something like that. I really found that interesting. Uh, okay, so we're going straight into the chloroplast, obviously, right? So large organelles, biconvex shape with a diameter of this, surrounded by an envelope, because it's double membrane, as with mitochondria as well. As explained here. Material is the stroma. Stroma contains small DNA and ribosomes. Flattened disc-shaped photosynthetic site called the thylakoids are present, and the thylakoids are arranged in stacks called grana. Okay, that much we all know. Over here, yeah, okay. So we have the stroma, we have the inner membrane, the outer membrane. We have no pores, but we have small amounts of DNA, as well as ribosomes, as well as these starch granules. Then we have vacuoles, I believe. These vacuoles. Oh no, those are the starch granules. Oops. Okay. So these are your starch granules. These are lipid globules. Interesting. Um, then the most important parts is we have your thylakoid. Um, you have your thylakoid membrane. You have your thylakoid like contents. You have your uh, uh, what are these connections called? Uh, I forgot what these connections are called. But anyway, each of these stacks of thylakoids are called your grana. Um, anything else? Do they say lamella, right? Lamella or lamellae, whatever. Um, Granal thylakoids and stromal thylakoids. So this is actually what they look like. Site of photosynthesis, as you know, chlorophyll pigments are found in the thylakoid membrane. Chlorophyll pigments are found in the thylakoid membrane. Um, yep, yep. So this proves that the photosystems are in the thylakoid membrane. Chlorophyll pigments trap light from the sun. Okay, why are we doing this here? Okay, no, we're not gonna go in. We're not gonna go in depth. Okay, I was like, are we gonna go into ATP synthase right now? I mean, not ATP synthase. Are we gonna go into Damn, I forgot what it's called. Are we gonna go into photosynthesis right now? Like the whole thing. I forgot that there were two cycles. The cyclic phosphorylation, non-cyclic phosphorylation. That's what I mean. Let's open it. Cause it's here. Might as well. Um, let's see. Where would this be? This would be in books. Uh, oh, not here. I should probably have. I should probably have this just right next to it. Do I not? I don't have study materials like pinned. Okay, let's do that. Don't mind this, okay? Uh oh. Uh. Oh. I messed up. Oh no. Okay, we're good. Uh. Where is it? This one? Was it 7A? Should be right. Yeah. Um. It was not. It was 5A. Are you serious? Really? Wow. That's crazy. Okay. It's not that crazy, but that's crazy. All right. Okay. Um. It's not easy. Um. So just how they explained. I really enjoyed how they explained the photosystem. So photosystem is a collection of chlorophyll, a primary pigment with accessory pigments and associated proteins all within the protein membrane, or the membrane protein, right? So it's all in that thylakoid membrane. Um, their role is to absorb energy from photons of light and excite electrons out of the photosystem in the electron transport chain. The reaction center of all photosystems contains chlorophyll A. um, there are two types of photosystems, photosystem 1 and photosystem 2. Both are used, the photosystem 2 comes first, photosystem 1 comes later. These are used in the non-cyclic phosphorylation, right? Whereas in the cyclic, I believe it's just PS2. They don't even mention it. Okay. No, it's only involves PS1. My fault. But the first one is PS2, right? Yeah. Okay. Yep. I wonder why. I need to read what they have because if they go crazy in depth, maybe I should ask her. Maybe she would know. But maybe she's not messaging me because she wants to focus on my work. That would be really funny. Cause like, okay, my fault. That's my fault. um, it's like actually my fault. That's really smart. Encouraging. Okay. 100, like let's do 150. I need to look at 118 and then so forth. 118 so forth. 118. What? Why are they talking about this? Wait, what? Okay, that's crazy. What the hell is this formula? Enthalpy, free energy, and entropy? Uh, okay, interesting. Okay, that's cool. This is unexpected. Let's open. Okay, I don't know if I'm actually allowed to show this, so let's just wait. I look beautiful. Sorry. Let's see. Entrance exam. Entrance. Ah, brr. Come on, come on. Turn the radio on. Sorry. Come on, come on. Turn the radio on. It's Friday night, and I won't be long. Come on, come on. It's Friday night, and I won't be long. And I won't be long. Where is the content, bro? Come on. Turn the radio on. Come, it's Friday night. I won't be long. Come, it's Friday night. Cause it's Friday night. I won't be long. Cause it's Friday night. Okay, I did not have to open this actually. That's my fault. That's my fault. I was looking at myself, by the way. I'm not. I'm not. I'm not. Let's see. Basic structure and characteristics of eukaryotic cell. Okay. Basic metabolic pathways. Glycolysis. What is glycolysis? Okay. Specifically, glycolysis, biological oxidation, photosynthesis, the biological role, and basic characteristics of enzymes. Okay, so none of this. Understood. Hmm. The role of ATP in biochemical energetics. Okay, listen, if I had time to study this, I would. Also, I probably wouldn't. I'm not good at reading. I'm not very good at reading. They're talking about activation energy. What the hell? This is like actually chemistry. This is so sick, man. What the hell? Okay. How enzymes, right, of course. Wait, what? Wait. Did they ask that? The basic characteristics of enzymes. Uh, Wait, can I place a bookmark here? Dude, how do I place a bookmark? Come on. Come on, game. Uh, it's here, isn't it? Boom. Enzyme characteristics. Okay, let's keep going. Oh, cool. Cofactors, coenzymes, prosthetic groups. Heme, flavin, retinol. What? Are you serious? Oh, okay. Okay, yeah. How am I gonna read all of this? Um, um, what the hell do you want me to do here? This is crazy. Okay. Um, okay, here we go. So this is an overview. But when we go into like the actual, okay, they're even doing the overview of those. Okay, nice. Oh my god. Am I supposed to know all of this? Oh my god. Oh my god. Did she study this? Is pyruvate oxidation the link reaction? Yeah, yeah, it is, right? Yeah. Wait, no. No, no. Pyruvate oxidation we studied was the end of the was the end of glycolysis. So it's different. Okay, I understand. Oh dear. I wonder, like, what do they actually expect? Okay, let's look at this thing. Okay, let's look at a key sample test. But let's go to the medicine one. Okay, what like what do they actually expect? Like, do they ask you like this kind of stuff, or like how in depth are we going here? Updated biology sample test. Okay, let's go. So, what is the function of saliva as a digestive fluid? Um, Carbohydrate digestion. Structures. Okay, these are fine. Okay, this is fine. This is 4 globin chains. That's true. Fe2 plus. That's true. Is produced in the circulating red blood cells. No, right? 23% of the if 23% of the bases in a sample of double-stranded DNA are adenine, what percentage of the bases are guanine? Uh, uh, was it is it ATCG or ACTG? It's adenine, then thymine, then cytosine, and guanine, right? So okay, so 23, and then that'll also be 23, so that'll be 46. 46 minus 100 will be uh 70, 46, 74. No, what? 54. Then 54 should be 27, I think. Okay, so far pretty easy. I'm sure it's gonna get a lot harder. Is it just? No, it isn't. Wait, what the hell? This is nothing compared to that. Okay, so I think I actually should be fine, right? If I just study from my notes for the most part. Okay, let's see. Let's just keep reading. So I don't know anything about Mendel's thing, but so the enzyme that restores the phosphodiester linkage between adjacent fragments in the lagging strand. I do not know this lagging strand thing. I do not know. I do not know that. Restores the phospho. Okay, but if it's restoring a phosphodiester linkage, what would it be? Would it be DNA ligase? What does DNA ligase do? No, that's not it. Primase, I'm not sure what that is. Reverse transcriptase turns an mRNA back into DNA. I'm pretty sure. Okay, 1 second. Cool to say. Yeah, you guys are you guys just think about it? Are you guys interested in that or what? Are you serious? Um, I understand. Okay. Okay, you don't need this. Okay. But I'm doing that very one. Oh. Shut up. Um. I've heard this one before. I forgot. Uh, okay. So like the Sensory Garden was my favorite. Oh. So then like what's it? But then I didn't really have. Oh, okay. Then I. Then I. Maybe it's this one. Okay, anyways. Do, 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 do. Are we good? There we go. Okay, what's up? So, enzyme that destroys the phosphodiester linkage between adjacent fragments and ligand strand. Okay, helicase that. Okay, wait, what is ligase? Ligase is the thing that reads it, right? Okay, my fault. Would that be it? Because that is actually used in DNA replication and it goes through. So that might be it. DNA polymerase 1. Wait, isn't that what it is? What is ligase? I have no idea what ligase is. Shit. What is DNA ligase? Helicase. Um, polymerase. Polymerase. RNA polymerase. Okay, well, okay, DNA polymerase 1 is so specific that I'm pretty sure it's this. But what is DNA ligase? What is DNA ligase? Acts as a molecular glue, joining DNA fragments together by catalyzing the formation of covalent phosphatase. When is it used? Okay, well, okay. To be fair, DNA polymerase reads. I don't reads and then forms the what's it called, the anti the anti strand or whatever it's called. Uh, so yeah, is the answer key here? Please, come on, don't do this. Key key. Please, no, not people. Bro, so annoying. Okay, there's also these. Can I open these? Do, 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 do. Oh, this is like an actual exam paper? This is bio? I don't know. This is, what the hell? This is bio? No, no it isn't. It's like all of them. Okay, here's one. Whoa, whoa, what? I'm so, where are the questions? Is this just notes? Hmm. Um, dude, this is PMO. Is there no test? It's true. Sorry. What the hell? Why is it? This is kind of annoying. Exam topics. Key sample test chosen. Key. Updated. Okay, fine. That's fine. Okay, let's go back to this. So I guess there isn't, which means I'm probably have to go through AI with it. So like, okay, let's. I just went back to the same one. I hate like how laggy this website is. Like it just is. It's so sad that it has to be like this. But anyway, okay, yeah. So highest degree of blood flow would obviously be in the smallest one, right? So the smaller arteries, or rather capillaries. Oh wait, but then aorta probably pumps with the highest pressure. Venules? No idea what that is. Is that like the the ventricles? Something like that? Because that would be a fun for sure. Could I like ask Gemini like through here? Answer all the MCQs. How does this work? Where were we? Okay, so the enzyme that restores the okay, yeah. So this one probably the zygos. I mean, I don't think it would be polymerase. Like I can't really tell you what the function is besides reading and making the antisense, right? Antisense strand for sure. Anyway, so in Mendel's bees, uh, tall and short plant variants resulted from differences at a single genetic locus, with tall being dominant to short. The differences between the tall and the short plants were caused by their different alleles and gene pools, alleles and genotypes, alleles. Since it's tall and short line variants, then probably LLs. Mitotic prophase, okay. Differs from prophase 1 of meiosis. Okay, cool. So they're going through that as well. Okay, what is this? I think they're going through some of the questions in the paper. Is it rain? I think it is. Not to be memorized. Okay, let's keep going. I mean, this is not really a big problem, because it's stuff that I know I can find in my notes. Which compounds link glycolysis in the citric acid cycle to the electron transport chain? Um, Glycolysis and the citric acid cycle to be, that would be NADH, right? No, no. Would it? It would be um, what is produced after the citric acid cycle? You release like loads of NADH. You release O2. You release CO2 probably. No, you don't release O2. You release CO2. You release NADH because you have like decarboxylation. You have dehydrogenation. But what are you left with? What you're left with is the initial thing, right? It's the initial thing that's used in the citric acid cycle. So then all that NADH and rather all that hydrogen, which was turned into NADH, I believe, which like reduces oxi yeah, yeah, which reduces NAD plus to NADH. Um, so then NADH is used. I don't know. And then I know that electron transfer chain starts with, what does it start with? It starts with an electron or something. It's so sad that I forgot this stuff, man. So over here, you are left with oxaloacetate. Okay, wait, hold on. Okay, yeah. Acetyl-CoA combines with oxaloacetate to form citrate. Citrate is decarboxylated. Okay, yeah. Okay. Yeah, so oxaloacetate is reformed, right? Regenerated. Okay, cool. Initial components are reduced NAD, so NADH and FADH. Okay, alhamdulillah. At least I have this. Okay. So it is NADH and FADH too. Cool. Naming was shown in the figures below, and the structures labeled by the numbers too. This is a, what on earth? Is that that's a prokaryote cell, right? I believe. So you have, um, I totally forgot what the outer, like the outer thing is. Let's turn off the because it's really not doing anything. That's like just cytoplasm. Those are ribosomes because they don't have a thingy. That is, uh, was it like nucleoid or like just circular DNA? Um, what is this called? Shit. I literally just learned it. Where's my thing? It is Oh, it's just called capsule. Slimy capsule. Yes, lamellae. Wait, we're learning about lamellae? Or okay. Whoa, that's cool. Nice. Okay, we're gonna. Oh, nice, 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 nice. I really wanted to study this. Okay, cool. Cell cycle and fertilization. Yeah, I'm super down because they're gonna talk about everything, right? The phases, nice. The DNA content, cool. Consequence of mitotic division, cytokinesis, meiosis, genetic variation, crossing over, independent assortment, how meiosis produces variation in gametes, cell like specialization or structure of the gametes. Similarities of the nucleus of human egg cell and the nucleus of a human sperm cell. Okay. Fertilization in flowering plants. I don't think that's there. Double fertilization. Yeah, no. Environmental influence on gene. Okay. Okay, so slimy capsule. Okay. Let's just keep going. Whoa. Okay, so this is I'm not really that good at it, but anyway, whether a pea seed is round or wrinkled is determined by a single gene called gene A. With round seeds, okay, round or wrinkled is determined by a single gene called gene A. So gene A has alleles which code for round or wrinkled. With round seeds being dominant, so it's the dominant allele. Wrinkled seeds are the wrinkled is the recessive allele. In the progeny of a test cross, I don't know what progeny means. 900 seeds were collected. How many should be round out of the 900 seeds if the unknown round-seeded plant being tested is heterozygous? Please give the genotypes of the test cross and the genotypes of the progeny as well. Okay, I do remember like there were lots of terms like genotypes. You need to know what the genotypes are, what the phenotypes are. So the phenotypes would be rounded and wrinkled, but the genotypes would be what? Would it be round and wrinkled as well? I'm confused. Um, and what does it mean by heterozygous? I do not remember. I do not remember. Genotypes of the progeny of the progeny of the testosterone. Since dash of the testosterone has this is such a okay. Yeah, I'm not sure. I have no idea. But okay, more MCQs. Okay. Um, what the hell is this? No, I have no idea. Wow. Oh God. Okay, now we're getting into it. So, partial pressure of oxygen is almost this in systemic arteries and this in systemic veins. Oh, okay. Yeah. There's one false statement involving the regulation of breathing. Choose one false statement. Which of the following takes place during the systole? Okay, that I know. Or I can learn. Progesterone. Okay, functions of the hormones. Okay, fine. Which protein is not part of the structure of a cilium? How would I know? Okay. Cilium was just the things like outside. It's not a prokaryote. That's flagellum. Cilium is just like the things on the outside of the animal eukaryote, right? So the statement enzymes are highly specific means that specific um, doesn't it mean like the binding or like the shape of the active site? Reaction with specific activation energies are catalyzed by specific enzymes. That sounds good. Reactions involving specific substrates are catalyzed by specific enzymes. I would say substrates, not activation energies. Concentrations of substrates work with specific enzymes. Um, no, it's specific substrates because like specific shapes of those substrates. Conversion of malate to oxaloacetate. Okay, in the citric acid cycle takes place with the conversion of NAD plus to NADH. In this reaction, NAD plus is N. Okay, this is a uh, NAD plus gets reduced, so it is an oxidizing agent, right? It's not inhibitor activator. I don't know. Oh god, so I'm supposed to know what those are because I remember seeing those in the textbook. Which of the following provides the correct order of events in the synthesis of the ligand strand? No idea. Like, absolutely no idea about this. A codon is this many nucleotides long. How am I supposed to know that? Okay. Wait, no, wait. A codon is 3 amino acids. 3 amino acids. No, not 3 amino acids. It's 3 nucleotide bases. So that would be 3 nucleotides long. And there are dash different possible codons in total. So it's B and E, 64 or 16. Probably 64. I mean, like I would imagine it's like 3 factorial. No, not sure how to calculate that. Scientists determined the mean diameter of muscle fibers in trained mice using an optical microscope to examine sections of muscle tissue. Circular area of one field of view was 1.25 millimeters squared. The diameter of this area was equal to the diameter of 15 muscle fibers. Calculate mean diameter of muscle fibers. So 1.25 divided by 15. This is 5 points? Okay, what? In micrometers. Okay. Oh, okay, and that's the circular area. Okay, sorry. So what's the order here, actually? This is a good question, because there's actually a lot of conversion going on here. So this is the diameter of this area was equals to 15. So then first we have to turn 1.25 into uh, a diameter. That diameter would be in millimeters. Then we would essentially, we could convert it first, but we would turn 1.25, or sorry, the diameter millimeter into divided by 15, and then turn that into micrometers. It's meter, decimeter, no, what is it? Meter, decimeter, centimeter, millimeter, uh, micrometer, nanometer. So that'll be like 1 times 10 to the power minus 1, 1 times 10 to the power minus 2, 1 times 10 to the power minus 3, and then micrometer is 1 times 10 to the power minus 6, and nanometer is 1 times 10 to the power minus 9, and the picometer is 1 times 10 to the power minus 12. Please describe the characteristics of the process shown. Okay, this is exactly that thing, right? Like, I have no idea. So like, I guess that's, what am I looking? Okay, so the figure represents DNA replication. The aim of the process is to replicate DNA. The figure represents what exactly? I mean, I'm not sure. The aim of the process is to like produce. Okay, but then you can see here that there's like two like, isn't it you just break off both of them? You make a antisense strand to one of them, and then you combine them, and then you have just one mRNA chain. Over here, I can see there's an, like, if we can consider this bottom thing an mRNA chain, now what is this on the top here? That's what I'm confused about. Ah, no, wait. But mRNA is for protein synthesis. What is it for DNA synthesis? Oh, bro, I totally forgot DNA replication. So if it's like this, then I imagine that like this bottom one and this top one combine them. So, um, oh wow. I mean, we did not learn. It's been so long since I studied DNA replication. Damn. Okay. So, um, I mean, the major enzyme required for the process is, I'm guessing, like DNA polymerase, right? The or like I know this is ligase. No, helicase, right? Ha, kuchi. Sorry. Okay. I don't really need to lock the door. I don't, I don't know, guys. I don't know. I'm kind of scared. But it's okay. It's okay. I mean, even if it doesn't go well, I want to have at least tried, right? That's the point. That's the point. I want to have given it my all, and if that was enough, then that was enough. And if it wasn't, then it wasn't, and it wasn't destined for me. So, and I'm completely fine with that. Okay, let's see. This I'm not so sure. This I'm not so sure. This we talked about this. Okay, so major enzyme required for this process, I'm guessing it's DNA polymerase. The dash strand is labeled by 4. So I'm guessing that's the leading, like, is it like the leading and the lagging? I don't remember. I think it's like, okay, but it's such a long thing. Maybe it's the leading strand. The function of the enzyme labeled by 2 is to synthesize the peptide bonds, or no, phosphodiester bonds, right? Here we have, wow, seriously. Okay, so not very comfortable with this either, with this topic. Damn, this perfume smells so good. The process, okay, wait, ovarian development, the female ovarian cycle. So this is completely new. Um, I don't, I don't even know what I'm looking at. Is this like a, is that an egg cell and then it gets fertilized? Is it? I'm not sure. Is the, no idea. Transport to the intrinsic membrane, don't know what that is. Facilitated, okay, which is active and which is facilitated. This is classic. Um, active requires ATP. What is facilitated diffusion again? Does that require ATP? I don't think so. But what was it? It was, I think it was like, it was basic, it was normal diffusion, uh, but using transport proteins because they were like too big or something to go through the membrane, something like that. So transport to the intrinsic membrane. Um, both, it's true for both. Um, requires ATP, false true. From high to low. So from high to low is just facilitated. That's the, really, that's it. That's it. Okay. Um, okay, buddy. Brody. There are no furries in Stalingrad. You know what's funny? Like that could be Azuma or it could be just a thought. Just a thought out there. Why am I scared? Oh, this is the 6-7 thing? Okay. Azuma wasn't in our class? What's this? Man, this guy just mogs me, man. Damn. Damn, bro. Okay, but yeah, I did see these photos. It's not. It's not that. Yeah, you're right. You're absolutely right, bro. Yeah, I'm not saying I'm not saying that like I'm trying to take care of you. Like no, but it's like um, it's going to happen, right? Like when you have a partner, it's not it's no longer at least when you start living together, whatever, right? I would imagine it's not just your thoughts in your brain anymore. It's like this person's presence is always there as well. So it's like you have a second brain inside your brain, and now you have to like so you know what I mean? It's not like I'm necessarily saying that. But yeah, she's absolutely right that I should not be stressing about this right now. Oh god, I'm such an idiot. Such an idiot. Fuck. God damn it. God damn it. Why am I doing this on camera? Uh, it's worth it. It's so worth it because if I do this on camera, then I can actually see myself next like sometime in the future, like when I'm like sad and depressed because it didn't work out, and I'll be like, you piece of shit. You should have studied harder. Okay. My fault. My fault. So what is all of this for? Is this pointless? I don't know. I don't know. I don't know. God, why am I stressing so much over something that's so pointless right now? Oh, that is so funny. That is so funny. That is so funny. Okay. Well, not anymore. Not anymore. No. Lock it. Lock it. Okay, so let's see. Basic structure of the eukaryote cell, basic metabolic pathways. Okay. Notice how none of those questions really went into that stuff. That is just one paper. I don't know. Okay, but like DNA came in a lot, a lot, a lot. Okay, so the process of DNA replication definitely not protein synthesis though. That's one thing, and its role in heredity. That's another important thing, right? So structure of DNA, genetic code, and the process of replication of the genetic material. Then cell division also came, and meiosis that was for sure that came. Then genetics, genotype, phenotype. Yeah, that was super important. Yeah. Monohybrid cross, dominant recessive, types inheritance, codominance, the first Mendelian law of inheritance. Okay, sure. Genetics 2, X-linked inheritance. Okay, I'll be, okay, so the point of focus is definitely here. From DNA to protein, oh, okay, this is also here. So they want us to focus on the translation part, not transcription. mRNA, rRNA, tRNA, ribosomes. Okay. What is rRNA? I have no idea what rRNA is. No idea. All I know is that it's in the nucleus. In the nucleolus. That's where it gets made or something. Basic anatomy and physiology. Okay, that was also there. Oops. Yeah, that was also there. A little bit of it, yeah. I want to go to the hormones part. So, like the ovarian cycle, those talking about like a lot of the hormones. Talking like referring to a lot about the hormones. By the way, wait, can you see it? Mango. I have a spoon while eating mangoes. Come on, that's so sad. Mmm, so good. I love Bangladesh. Okay. This is the best idea. I don't think like I think this is the best thing I've ever done. Turning on my camera. Hopefully, not too many people watch. Cook this in. Oh my god. What happened? Sorry. Um, there are some topics where there's some stuff that I don't know. For sure. I mean, mock exam to complete it. Or mock exam offer করছে তো আর দিতে হবে।
দিয়ে দাও তুমি শেষ করো করে কালকে সকালবেলা বলে দিও তুমি মকের জন্য রেডি হও রেডি হয়ে তারপর উঠতে চাও ঠিক আছে you will not get much time by the way so একদমই টাইম
I'm just saying এটা বায়োলজির জন্য এখন কেমিস্ট্রি
হ্যাঁ তো এটা একটাই আগে শেষ করো পুরো সাবজেক্ট ওয়াইজ দিবে আর যদি তাহলে এটা এই পরেরটা নাও ফার্স্ট
জানুয়ারি
না জানি না জানুয়ারি না
Do you think it'll be in July or do you think it'll be in this month?
কি আছে কিন্তু ওকে আমি বলছি যে জুলাই যাব আমরা তখন ও বলল যে ওকে দেন I will if the date is they give earlier then I will talk to him and manage but যেহেতু ও বলছে যে ও ম্যানেজ করবে বলে যে না ম্যানেজ করতে পারিনি then অনলাইনও হয় আবার ইয়েতেও হয়, ডিপেন্ডস অন কি জন্য করবো, যে কতজন ক্যান্ডিডেট হচ্ছে এটার বিষয়ে অনলাইন হয় আবার face to face।
Sure. Um, uh, so, she just told me that her parents didn't specifically say no, but she also says that uh, oh yeah, wait, she just told me. She said she really wants me to focus on the exams, which is like funny, but yeah. Anyway, so she
Why is it funny?
it's funny because I'm not doing that. I'm stressing over this stuff, and she's telling me don't stress over this stuff, and
No, why do you stress over this
because
stuff?
I'm human. That's why. Okay.
No.
Okay, I'm sorry. Um, she said it puts her mind at ease. My parents did not say no. They both want to discuss with both of your parents in person. Once your father knows anyway. Once your father knows.
Really?
Yeah.
It's not.
Yeah.
That's not what you say. Then why do you stress it?
I'm not stressed. Okay. Shoo. I'm sorry. I'm sorry. She said she won't talk to her on this. She. Can I just not? Okay. I'll tell you that we're coming back by.
And he's feeling যদি এই অবস্থায় আসে আর তোমাকে আমি তোমার পরীক্ষা এসব নিয়ে তুমি নিয়ে আমি চাচ্ছি না তুমি এখানেই so এখন শুরু করো
She said she wants to. She just said she wants to. The parents want to talk to her. Okay, I mean, if you tell them our circumstances, then always open.
এগুলো নিয়ে এখন আর রিসার্চ করা
Okay. Yeah. I should just focus on my
পরে
phone.
যাচ্ছি এগুলো নিয়ে কোনো কিছুই জানো না তুমি এখনো তো এখন সবাই তোমার এক্সামের উপরে
Yeah. Yeah, sure. Part two, honestly, just for to get a date. No, I'm forcing. I'm using I'm not using it. Okay,
শোনো
done.
আমি তোমাকে বলি গিভ ইট টু মি জাস্ট এখন কম্পিউটারে অন করো না
No, I'm gonna just put that.
একদম তুমি এটাকে সাইলেন্ট করে দিয়ে একটা ফিক্স করো যে আমি 3 ঘন্টা আমি কোনো মেসেজ দেখবো না সেটা যদি 3 ঘন্টা লেটে দেখো আর যদি পরে বলো যে আমি এটা এখন বন্ধ রাখছি 3 ঘন্টা জাস্ট বসছি এটা আমি চেক করে নিই it is very much very important
Yeah, I'm sorry. I know. I locked it. Oh my god. Ooh, my days. Oh, my days. Oh, my days. Oh, my days. Oh, my days. Can I like increase? Whoa, that's a. Okay. Uh, wait, what if I did custom? Nothing, nothing, nothing. Wait, why is my hair doing this? I don't know. It always does this when it gets long. It's so annoying. I look like an idiot. I look like a freaking idiot. Okay, just forget it. I look. It's over, bro. It's so over. Okay, this is talking about hormonal stuff, right? It is literally ovarian cycle. Okay, so uh, does this talk about everything? Endomembrane system, Golgi apparatus, mitochondrial structure. The previous exam did not talk about breathing. I should really ask her like on tips and stuff. Oh wait, let's look at talk about Uni of Debrecen entrance. Yeah. Okay. I should see in like 5 minutes. Seen a lot of people say the biology part is easy, while the topic list was given was topics that was easy. The exam was very tough for biology. I'm saying this because I do ethics. Okay, so this guy is basically me. And our biology isn't that detailed. I thought it was only me who had a hard time doing the biology exam written part B, but it looks like other candidates who are appearing to have some difficulty. The questions in the biology written part were based on a pedigree diagram and Mendel's law, which was easy. Then I asked some in-depth questions about DNA. I don't remember pretty well in those questions. They used words that I believe only a first-year med student would be aware of. Mind you, I'm 17, will turn 18, and with me, other candidates were 20, 21, so I believe they had better knowledge than me, as they may have taken greater to prepare. I don't know. Some girls had taken Canadian and American board, weren't able to do the bio written part too. So if you're 17, 18, there will be people older than you. In my case, it definitely was. I only knew 3 other girls who were okay, I think this is a girl. Now, what I took from this exam was that be very well prepared for the DNA and Mendel's law. For chemistry, you also needed to know about intermolecular forces, esters, organic reactions, and how to name products. Also, acid, bases, pH, BOH stuff, as well as KC. Oh my god. Okay. As for the oral part, examiner is very chill. If you don't know anything, you just tell them. In my case, I had 2 professors. One was female interviewing about chemistry, and one was male professor interviewing about biology. They first asked, "Tell us about yourself," and I prepared. I had material prepared from ChatGPT. I had to be very honest because these professors love honesty. I told them how I love dancing and singing. They had big smiles on their faces and asked me a question if I play music. This was asked me, which I replied, I sent a little bit. Then they started. They let you choose which subject you want to begin with. I chose chemistry, and she was a bit timid, not gonna lie. She asked me about. I'm glad she's just like telling this stuff. This is so cool. Okay. Um, she asked me about mixture solution concentrations unit. Um, she asked about cycloalkane, so no organic from the inside out. I told her my school didn't really focus on organic much, so I don't know in detail. Even though my school did, I was just bad at organic. But if you're getting organic, I believe you'll be good to go. Then we started with biology. He asked me about homeostasis. I explained to him with many examples in case of dehydration, etc. What? Oh, what? Oh, okay. I mean, okay, sure. I was going to explain with blood sugar. Yeah, I kept on explaining. He didn't stop me once. Only when I stopped did he proceed to the next. He asked me about the stomach, and I told him it was 1.5 to 2 due to the HCl, and I told him HCl helps in protein digestion. Mm, I mean, isn't it like you want the optimal pH for the enzymes? He was like, how so? I knew the answer. I explained that stomach chief cells, inactive pepsinogen. Uh-oh. He asked about lipid digestion, so I told him bile emulsification lipase. He asked me the source of lipases, which I didn't know, but apparently it's the pancreas. He asked about body temp, whether 37 degrees is internal or external. It's internal. He asked me about lungs and what happens to pressure when we inhale. I explained adding Boyle's law, and I stated extra info that I knew. He seemed to be very impressed with that. He asked about thyroid, something I told him it wasn't really taught to us. Keep in mind, I wasn't able to do that well in chemistry, but I made sure to impress my professor during the biology part. Anyway, me and alongside almost 45 out of 50 students got BMC. Wait. Shit. Is BMC the foundation thing? Wait, I'll need to check.
I don't know what they mean. আবার soup বানাইছে মানে ওই দুই দিক থেকে খাবার অর্ডার দিছে ডাবল হয়েছে
Okay, alhamdulillah. My interview went pretty well as much as I thought. However, I also got into BMC. I can't comprehend why. So I'm retaking my exam because I know I have potential. I have 3 A stars. So she got in like without a BMC. They're putting everyone in BMC. Honestly, it's very rigged because even after BMC, there's no guarantee you'll get into MD. Wait, what? Hmm. Well. Okay. Okay, fine. Since I'm going for that anyway, like, I should be okay. Oh, God, I really hope so. Oh. Oh my God. Okay, so, bro, I'm so good. Okay, shut, shut. But the fact that she got into the program must mean she studied pretty well. Okay, which is great. I mean, which is great. Which is fine. Which is fine. Yep, that's fine. Yep. Okay. I'm stressing again. I don't know why. I'm like, it's completely fine. Okay. What about say? I think it's like really stupid to say that I have a passion in biology, but then I like know nothing about biology. Like maybe it's like at most, it's like I have a passion for like, or it's not even a passion, but it's just I enjoy studying biology. Or I love going down rabbit holes when studying biology, but I don't like studying. I don't like actually the process. But I sometimes do. I don't know. It's weird. So it feels always like kind of like hypocritical or stupid when I say like I have a passion for biology, but then it's like I don't know jack shit. Like, and there are people who are like who just study harder and they know. It's like if anything, they have a passion for biology because they actually know or they put in the work. Um, I just have a very casual relationship with psycho with biology. I would say that. Like, I'm not very hardcore. Um, G. Okay, I'm gonna probably stop the stream because I think this is enough. It's 1 hour 50 minutes. I'll just study on my own, but this is really fun, and I'll probably do